Welcome to G Fun Facts Online. Imagine holding a time capsule, not made of metal, but of ice and earth. Inside, life from a forgotten world, a world of woolly mammoths and saber-toothed cats, has been waiting. Today, we're not talking about fossilized bones. We're talking about unearthing living organisms from the Pleistocene epoch and even earlier, waking them from a slumber that has lasted for tens of thousands, even millions of years. This isn't science fiction. In the frozen landscapes of the Arctic and under the crushing pressure of the deep sea, scientists are reviving ancient microbes. This is a journey into our planet's deep past, one that holds both incredible promise and potential peril for our future. So where are these ancient life forms hiding? The key to their remarkable preservation lies in Earth's natural freezers, permafrost, and deep-sea sediments. Let's start with permafrost. This is ground that has been continuously frozen for at least 2 years, covering about a quarter of the land in the northern hemisphere. It's a mix of soil, rock, and ice that can be hundreds of meters deep, and it contains a staggering amount of organic carbon, about twice the amount currently in our atmosphere. For a long time, scientists thought that any microbes in permafrost were just dead cells, frozen relics. But research has shown that permafrost is home to diverse and viable microbial communities. The constant sub-zero temperatures, lack of oxygen, and low water availability create an environment that's incredibly effective at preserving biological material. It slows down metabolic activity to an absolute crawl, inducing a state of dormancy. Some microbes even form spores, which are like tiny, armored survival pods that can withstand extreme conditions for geologic timescales. It's a form of natural cryopreservation, an arc of ancient life frozen in time. Then there's the other repository of ancient life, the deep sea floor. In places like the South Pacific Gyre, where nutrients are incredibly scarce, scientists have found microbes in sediment layers dating back over 100 million years. These organisms exist in a state of near starvation, with energy levels millions of times lower than their cousins on the surface. The immense pressure, total darkness, and nutrient-poor conditions create another unique environment for long-term survival. These microbes are in a state of suspended animation, with metabolic rates so slow they're just barely alive, doing just enough to maintain their cellular integrity over eons. It seems that tiny traces of oxygen in these ancient sediments are a key factor in their survival. So we have these natural freezers, one cold and one high pressure, both holding on to life from a time long past. But how do scientists actually wake these sleeping microbes? The process is delicate, painstaking, and requires extreme precision. It all begins with collecting samples. For permafrost, scientists drilled deep into the frozen ground to extract ice cores. For deep-sea microbes, they use specialized drilling ships to pull up sediment cores from beneath the ocean floor. The single biggest challenge here is preventing contamination. The modern world is teeming with microbes on the drilling equipment, in the air, on the scientists themselves. To get a pure, ancient sample, researchers use stringent, aseptic measures. For instance, when they have an ice core, they often melt and discard the outer layers, using only the pristine, untouched interior for analysis. Once the sample is safely in the lab, it's carefully thawed and placed in a nutrient-rich medium, a kind of microbial feast. The goal is to coax the organisms out of their dormant state by giving them an abundance of food and a comfortable temperature, which for an Arctic microbe might feel like a heat wave. The revival process can be incredibly slow. In one study, microbes from Pleistocene permafrost showed very little growth for the first few months. After about 6 months, however, things started to happen. The community changed dramatically, with some bacterial colonies growing into visible biofilms. Reviving ancient viruses is a bit different. Viruses are parasites. They need a host cell to replicate. So to wake them up, scientists expose the permafrost samples to single-celled organisms like amoebas. If the amoebas start dying, It's a clear sign that a virus has become active and is replicating. But simply reviving a microbe isn't enough. To truly understand it, scientists turn to paleogenetics, the study of ancient DNA, or aDNA. This is where they read the blueprints of ancient life. Working with aDNA is extremely challenging. Over tens of thousands of years, DNA breaks down and fragments into tiny pieces. It's like trying to reassemble a shredded book. The samples are also heavily contaminated with modern DNA, and there's very little of the original ancient DNA to begin with. To tackle this, paleogeneticists work in ultra-clean laboratories, use special enzymes to repair damaged DNA, and employ powerful computers to piece together the fragmented genetic code. The revolution in this field came with next-generation sequencing, or NGS. These technologies allow scientists to sequence millions of DNA fragments at once, making it possible to reconstruct entire genomes from highly degraded samples. This genetic analysis is what tells us who these microbes are, how they evolved, and what they might be capable of. The history of microbial resurrection is filled with some truly mind-boggling case studies. In 2018, scientists in Siberia made a stunning discovery. They found 2 species of prehistoric nematodes, or roundworms, that had been frozen in permafrost for about 42,000 years. After being carefully thawed, these multicellular animals began to move and eat, officially becoming the oldest living animals on Earth. This proved that even complex life could survive for tens of thousands of years in suspended animation. But we can go back even further. In the year 2000, researchers announced the revival of a bacterium that had been dormant for an incredible 250 million years. This microbe, a species of bacillus, was found trapped in a tiny fluid bubble inside a salt crystal from a mine in New Mexico. The crystal formed during the Permian period, a time before the dinosaurs, when all the continents were fused into the supercontinent Pangaea. Reviving a life form from that era provided powerful evidence that life can survive on geological timescales. Then, in 2020, a team revived microbes from deep-sea sediment that was over 100 million years old. These organisms had been in a near-lifeless state since the age of the dinosaurs. When provided with nutrients in the lab, almost all of them responded, starting to eat and multiply. This discovery pushed the known limits of life, showing that organisms can survive for immense periods with virtually no energy. And of course, there are the headliners. The